Good morning, children. So today we are going to see regarding the continuation of the chapter Principles of Inheritance and Variation. So today we are going to cover sickle cell anemia and phenylketonuria under Mendelian disorders. First, let us see regarding sickle cell anemia. In case of sickle cell anemia, the people know suffering from this problem, they are showing a particular malformation of the amino acid. So what type of problem is there? Let us analyze. So here the glutamic acid, it is replaced by valine in the people suffering from sickle cell anemia. Actually, hemoglobin, it has two chains, alpha chain and beta chain. In the case of beta chain, no, the sixth position, the glutamic acid, it is changed into valine. So why it has been converted? Because the amino acid substitution it takes place. Why is it like that? Because it is due to single base substitution. So means in the case of GAG, the GUG change occurs. So instead of A means the U will be there. So because of that, the normal people, they will be producing glutamic acid, but here the sickle cell anemic people will be producing valine. So because of the change in the single base. Okay, so because of that, the people, they will be producing the shape of the RBC will be little bit changed. It will be sickle shaped. Because of that, the people also will be suffering from oxygen carrying capacity, because the cell is of sickle shape, so the oxygen carrying capacity, the WBC also will be less. Okay, so the people, how is it being transmitted, the disease? The disease is transmitted by a gene that we denote as HBS. So the normal people, they will be having the gene HBA, and the people suffering from sickle cell anemia will be having the gene HBS. So here HB is for hemoglobin, A we are denoting as for normal people, S we are denoting for sickle cell anemia. HBA means the people, they are fully normal. HBS means they will be suffering from sickle cell anemia, and HBS if they have like that, so they will be heterozygous for the particular gene. No, one will be normal, other will be sickle cell anemic. These people, they will be normal, but so they will be the carrier of a sickle cell gene. Okay, HBS are the carrier of sickle cell gene. They won't be showing the problem, but they will be carrying the gene, so that's why they can inherit the gene in the future generation. Okay, so HBA they are normal homozygous people, and HBS they are homozygous sickle cell anemic people. HBA and HBS, this type of genotype, it is there, they are heterozygous carrier people. Okay, so they are having 50% probability of transmission of gene. Why? Because they are having 50% of the gene, it is HBA, and next 50% is HBS. So now let us see how is it being inherited. So now let us form one Punnett Square. Here the HBS people, they are carrier. Okay, so they are able to produce two types of gametes. So one type will be HBA, other will be HBS. Means HBA is normal and HBS is sickle cell anemic gamete. Okay, so the other, this is also once again heterozygous people, so they are also producing two type of gametes. So one is female, this is male, and this is female. So actually this inheritance is autosomal inheritance. So why is it autosomal? Because the disease carrying gene is present in autosome. Autosome means body feature determining chromosome. Okay, so the previous one what we studied, hemophilia, it is sex linked because it present in X chromosome or sex chromosome. But here the disease causing gene is present in autosome. So that's why you call that as autosomal disease, and once again it is recessive. So that's why you call this disease as autosomal recessive disease. Okay, so here 50% of the gametes produced by male will be normal. The same way 50% of the gametes produced by female also will be normal. If we plot in Punnett Square, HBA, HBS, these are the gametes produced by female, and this HBA, HBS that is produced by males. So they produce the offsprings of different types. You see in the first square, HBA, HBA, normal people, HBA, HBS, that is no heterozygous, HBS, HBS, sickle cell anemic people. So means the produced is no like Mendelian, this one Mendelian type of no inheritance, 1:2:1, the F2 generation. In the case of monohybrid cross, the same way it is producing the same type of genotype. Okay, the phenotype will be, so the same way the normal people, when you see fully normal, that will be 25%, carrier people will be 50%, and 25% will be of sickle cell anemic person. Okay, so this is the type of inheritance in sickle cell anemia. So next we will see regarding phenylketonuria. This is also autosomal recessive type, the same way like sickle cell anemia. It is also autosomal recessive. This is also the same way inborn error, metabolic problem, but the problem is different. So here the affected people, so they are normally, when you observe, the normal people will be having the enzyme phenylalanine hydroxylase. What does it do? The phenylalanine hydroxylate, it converts phenylalanine to tyrosine. In normal people it is present, but the affected people, they won't be having that enzyme. Which enzyme? Phenylalanine hydroxylase won't be there. If it is not there, the people, they are not having the ability to convert phenylalanine to tyrosine. They won't be able to convert. So in the absence of the enzyme, what happens? The phenylpyruvic acid, okay, and other derivatives are produced, so by the phenylalanine, because phenylalanine, as it is not able to be converted into tyrosine, they are converted into some other substances. So accumulation of these type of other derivatives, it causes mental retardation, and the mental retardation, and also once again the kidney problems. So why the kidney problem occur? Because any poisonous or toxic substances are accumulated, it has to be excreted through kidney. As it is being excreted through kidney, once again people, they can suffer from kidney problem also, and the urine also will be having that phenyl pyruvic acid and other derivatives. If you see that type substances in urine, we can tell the people is suffering from phenylketonuria. Okay children, so today we studied two types of diseases. So one is sickle cell anemia, other is phenylketonuria. These two are autosomal recessive trait because the genes are present in autosome. That's why we call that as autosomal disease. As it is recessive, it is a recessive gene, we call that as autosomal recessive trait. Okay, hope children you understood these two. Thank you.